A question that many biologists find really fascinating is that we develop as a multicellular organism containing about 200 different cell types that have very different morphologies and physiology occurring, while each of them receives the same genetic instructions, which are our DNA, which is also called the genomes. So this is only possible because the different cell type need to express, that means to activate different genes within the same genome. As an analogy, we could think of our genome as a huge library, and the books within this library correspond to our genes. So if you want to become a medical doctor, you need to find and read books in this library, the medical books in this library, whereas if you would like to become a lawyer, you're going to read the law books within the same library. So similarly, a skin cell and a heart muscle cell will need to read different genes within the same genome. And this process of reading genes is called transcription. Now, of course, this analogy isn't perfect, and one of the differences is that books are physical objects which you can remove from the library, whereas genes are actually just segments within a long molecule of DNA present in our cell, and they are physically connected with the remainder of the genome and chemically really almost identical to the DNA that surrounds them. Also, genes can be read from the left to the right and from the right to the left, so they can be read in the forward and the reverse orientation. So to visualize this differently, I depicted the same three genes here as boxes, and the direction in which they're read as arrows. Of course, this is a huge oversimplification because, as Wojtek just told you, genes contain introns and exons and things that are, of course, a bit more complex. Nevertheless, much scientific effort has been put into finding out how the cellular machinery manages to distinguish, to find the beginning of the gene and start reading into the gene. However, we know way less how to stop reading a gene. So this process called transcription termination is important because it punctuates the ends of the genes, and it also is really important to release both the transcription machinery and the products. But also in the recent years, we realized that the non-genic material, the non-coding parts of the genes are also very heavily transcribed. So it's also very important to control this non-coding transcription. Now, in the transcription fields, as well as many other fields of biology, a lot of what we know is derived from studies of very simple organisms, model organisms, such as, for example, yeast. And the studies from many groups working in yeast have discovered that one critical termination factor that is also important to link transcription with other processes occurring in the nuclear is called PCF11. Sorry, went a bit too fast here. So in my recent research, I really wanted to find out what is the role of this critical factor, but in vertebrates. So my main model has been human cells that I grow in the lab in plastic dishes, but also I confirmed and extended some of the finding using the zebrafish animal model. And the research approach that I'm taking has been to modulate the levels of this PCF11 factor and then checking how this affects transcription by using techniques that are derived from next generation sequencing that I enumerated here. So, so in this slide, I'm summarizing what we have found, which is that that invertebrate PCF11 preferentially binds and acts on genes that have a close-by neighbor. And this is very important to punctuate those genes and prevent the transcription from running from one gene into another, which interferes with the reading of this neighboring gene. So this is something that positively impacts transcription. But what was actually very surprising to us that we realized that there's a subset of genes where PCF11 is doing a weird thing. It's actually prematurely stopping transcription before it reaches the end of the gene. So this, of course, leads to downregulation of such genes. They cannot be as active anymore. Okay, so it turns out that those genes are not just some kind of random genes. There's a tendency, they tend to be associated. They tend to encode factors that are important to regulate transcription and also other aspects of gene expression. And this is true both in human cell culture, but also in developing fish. And in fact, in many cases, exactly the same gene is prematurely terminated in human and in fish. And this for a biologist is a very strong indication that this is likely to be, because it's evolutionarily conserved, it's likely to be an important regulatory process. Now, interestingly, I found that one of the genes that is prematurely terminated by PCF11 is the gene that encodes this factor itself. So what my experiments have shown is that when the levels of PCF11 are high, these genes get prematurely stopped. The reading of the gene gets prematurely stopped, and this leads to lowering the level of this factor. But then when the levels of PCF11 are low, then the reading goes throughout the whole transcription unit. And I've also could show that when I delete the genetic element that is important for this premature termination, I get more transcription through the gene and a higher level of this factor. So what is going on is that this factor is able to auto-regulate its own levels. And this regulation ensures that the cell has stable levels of PCF11, and also those levels are actually relatively low. So fine, so the cell is putting a lot of effort into getting the right amounts of a factor, but does it really matter to a living organism? And the answer is yes, because when we deplete PCF11 in zebrafish, they die very early during development due to brain necrosis. And also, if we provide zebrafish with too much PCF11, they again develop another range of developmental abnormalities, for example, shorter larvae length or distinct tails. So really, for an animal to develop properly, you do need to have just the right amount of PCF11. Okay, so we have this factor that is very tightly regulated. This regulation also seems to be important in animal development, but you know, maybe somebody could ask, does this have any significance to human health? And until very recently, there were no known diseases that would be associated with PCF11 malfunction. But just in the few last months, 3 independent screens for driver mutations in cancer have found that there's actually frequent mutations occurring in the promoter region of this gene. So this is the region that can actually determine whether there's more or less transcription of the gene going on. Actually in just this month, a really beautiful work from Sven Dankert lab has been published as a preprint that shows direct role of PCF11 in a pediatric cancer which is called neuroblastoma. And in fact, in this type of cancer, the level of PCF11 is very highly predictive of the clinical outcome. So further studies of transcription termination and this critical factor, they are going to not only help us understand how genes work, but they can really have also medical implications. So the take-home message that I would like to leave you with is today is that transcription termination is a fundamental process which occurs in every cell of our body. And also that it really matters to know when to stop transcription. With that, I would really like to thank my current boss, Nick Proudfoot, and my collaborators who are listed here, and also acknowledge funding from Mary Skłodowska-Curie Actions that funded a big part of my postdoc. And I also would like to do some advertising, because as Kasia mentioned, I will be actually moving to Poland shortly. I will be setting up my lab at Adam Mickiewicz University in Poznań. So the lab will be affiliated with the Faculty of Biology and physically be located within this beautiful brand new building of the Wielkopolska Center of Advanced Technologies. And actually, just today, I received official information about having been awarded a grant from NAWA within the Polish Returns program. So this allows me to employ 2 people, 2 lab members. It's quite a European, I think, competitive salary. So the official recruitment will only start next year, but please spread the word, or if anybody's interested, please contact me for more information. And I'll maybe also mention that I do quite a lot of computational work, so I'm both open for people with biology background, but also quantitative computational background as well. For more senior people in the audience that are thinking about starting their own research group, I can also really recommend considering Poznań, which especially in the RNA research community is very strong, which was recognized by awarding it the status of this leading national research center. And I have also to say that the faculty is actually increasingly international, not only at level, not only as far as the students are concerned, but we also have a few group leaders which are non-Polish nationals. So I think it's really a great place to do science. And I stop here. Thank you for your attention. I'll be happy to take any questions.